Welcome to Unit 2, Macromolecules and Enzymes Review. You should be able to recall some of the elements found on this table. We're going to put a lot of emphasis on carbon. Carbon makes up 18.5% of your human body. The rest of the elements on here make it into your body through your diet. So what makes carbon so unique? Well, carbon can bond to 4 additional atoms thanks to its 4 valence electrons. Carbon molecules have strength, flexibility, and great versatility to chemically react with other atoms' molecules. Carbon can make very long molecules, and this is a property that no other element even comes close. So molecules that contain carbon are known as organic molecules. Complex organic compounds are known as macromolecules. Macromolecules can be subdivided into 4 categories. We're going to be talking about carbohydrates, lipids, nucleic acids, and proteins. Dehydration synthesis is the process that creates these long chains of carbon. They join monomers into polymers. Hydrolysis would be the opposite. Notice that hydrolysis needs water in order to break bonds, while dehydration synthesis, or also known as condensation, will need to get rid of water in order to bond 2 monomers together. Now let's take a look at carbohydrates. Carbohydrates are made of carbon, hydrogen, and oxygen. They form ring structures. Their function is mainly to provide the cell with energy. However, they are also known to build plant structures and other structures in animals. Examples would include glucose, sucrose, glycogen, starch, and cellulose. The building blocks for carbohydrates is glucose. The chemical formula for glucose is C6H12O6. Glucose is usually found in the blood. Now, when glucose is present in the blood, the pancreas will release insulin. The insulin will attach to the cell and will let glucose into the cell. This is very important because glucose is used as the energy source for cells. Glucose comes from food that contains carbohydrates. Examples of those would be sugar, rice, pasta, bread, and cakes. Carbohydrates can be subdivided into 3 categories according to their structures. Monosaccharides would be the 1 sugar unit. Disaccharides would include 2 sugar units. These are simple sugars. Polysaccharides are the polymers. They include many sugar units. An example of monosaccharides would be glucose and fructose. An example of disaccharides would be the table sugar, also known as sucrose. Starch is an example of a polysaccharide only found in plants. It's used for energy storage. Cellulose is also only found in plants. In animals, we have glycogen. It's used for energy storage. And chitin is used to make exoskeleton. This is an example of how chitin makes up the exoskeletons of certain animals. Now let's take a look at lipids. Lipids. Now lipids are made of 2 components. We have a glycerol backbone and 3 fatty acids linked to the glycerol backbone. They're nonpolar molecules, and that's why they cannot mix with water. They are made of carbon, hydrogen, and oxygen, and they make long chains. Some of the functions include insulation, energy storage, and building cell membranes. Examples would be wax, fats, oil, phospholipids, which are found in cell membrane, and steroids. Now, examples of steroids include hormones and cholesterol. Steroids have a 4-ring structure. This is very different from the fatty acids and glycerol structure that we talked about earlier today. There's good and bad cholesterol. The bad cholesterol will stick to your veins and arteries. However, the good cholesterol will be found in cell membranes. Cell membranes are flexible because of these cholesterol molecules in it. Let's move on to nucleic acids. Nucleic acids are made of carbon, hydrogen, oxygen, nitrogen, and phosphorus. They make up single or double-stranded structures. Their function includes holding and transferring genetic information and also providing the cell with the codes for making proteins. An example of a double-stranded nucleic acid would be DNA, and the single-stranded molecule would be RNA. The building blocks for nucleic acid would be nucleotides. Nucleotides can be made of phosphate, the sugar, deoxyribose sugar in DNA, and a base. DNA is a double-stranded molecule, and that's the one on the right side, and RNA is the one on the left side. Notice that all the bases are the same, except for the last one. RNA contains uracil, and DNA contains thymine. We are going to be talking more about them when we get to the last unit in this marking period. This is just a quick diagram showing the difference in nucleotides found in DNA. Let's move on to proteins now. Proteins are very, very important to your body. They make up your hair, your nails, blood, enzymes, and other parts as well. Proteins are made of carbon, hydrogen, oxygen, and nitrogen. Their functions vary a lot. They include fighting diseases, making up structures, transporting molecules, and speeding up chemical reactions. Some examples include antibodies, hemoglobin, and enzymes. Proteins are made of amino acids. Amino acids are the monomers or building blocks of proteins. All amino acids share certain things in common. The carboxylic acid group will be the same in all the amino acids that we see, and the amino group as well. The only thing that changes from one amino acid to the next would be the side chain. We also know the side chain as the R group. This is the only thing that gets replaced from one amino acid to the next. This is just an example of the 20 essential amino acids. You do not need to know them. You do not need to draw them either. You just need to know that there are 20 essential amino acids that can link together to make proteins. This is just an example of how complex a protein is. It has to fold in a certain way in order to be functional. Let's talk now about enzymes. Now, enzymes are very important because, among many other things, they help us digest the food that we eat. We have different enzymes for different types of foods. Enzymes are a very special protein. Enzymes speed up a reaction by providing an active site for substrates to react. They help weaken bonds, and they lower the activation energy that the cell needs for a chemical reaction to start. Notice that enzymes are not destroyed or changed by the chemical reaction. They can actually be reused after the chemical reaction is done. Substrates is just another word for reactants. Just picture activation energy as the energy needed for something to get started. If you take a look at this diagram, this shows how a reaction takes place in the presence and absence of enzymes. If enzymes are present, cells require less energy to activate a chemical reaction. Enzymes like to work in a certain environment. For example, enzymes that belong to human cells like a temperature of 37 degrees Celsius. Enzymes that belong to the human body also prefer a pH between 7 and 8. Not all enzymes are created equal. By that I mean that some enzymes will rather work better at a temperature close to 37 to 40 degrees Celsius, while others, like the hot spring bacterium enzymes, will rather work at 70 degrees Celsius. This is also true for the pH. Rate of reaction means how much work the enzyme can do in a certain period of time. This is also referred as the enzyme activity. Remember, enzymes speed up chemical reactions. This means that it takes less time to produce a product. In this example, the pH of 6 is the ideal pH. It took less time to complete the hydrolysis, meaning that the enzyme was very, very active and was able to produce product in a timely manner. Well, how does an enzyme really work? Well, notice that the enzyme is very particular of the substrate. They have to match. Once they bind together, they are called the enzyme substrate complex. Once the substrate is attached to the enzyme, the bonds will be weakened by the enzyme and will break. The products are released and the enzyme is free to bind to other substrates. Notice that the enzyme is not consumed or changed in any way by this chemical reaction. The opposite is also true for enzymes. Enzymes can take two substrates and help bind them together and then release them as a product. The lock and key model of the induced fit model is just a way to describe how the enzyme and substrate work. The key would be the substrate, the lock would be the enzyme, and it has to be a perfect match to work. So remember, enzymes are a type of protein. So what can make an enzyme inactive or just make it not work properly? Well, things like changing the pH, the salt concentration, and temperature can make the enzyme unfold, change its shape, and make it inactive. Just a quick example of this would be when you cook an egg. Notice that the proteins are nicely folded together in the beginning, and then as you add heat, you change the shape of the protein. This is an example of how an enzyme can be broken or denatured or just made inactive. Well, this is the end of Unit 2 review. I hope I have clarified a lot of your questions. Let me know if you have any others. Thank you and see you in class. Bye.